Hi guys, this is Dr. Cummings from Point Loma Nazarene University. This is the third video in a 5-part series on the biochemistry of photosynthesis. I would not recommend that you try to tackle this without having watched the first two videos, Overview Part 1 and Part 2, to make sure you have context for all of this, because we're going to get into some really nitty-gritty details. Now, photosynthesis and its biochemistry can be broken into two broad sets of reactions, what we call the light reactions and the dark reactions. The light reactions, as the name implies, require light, and the light reactions actually themselves have two parts, what we call non-cyclic and cyclic electron flow, and they generate two different things. We're going to see the value in both of them, and so we've got a separate video for each of those portions of the light reactions. And then we'll finish with a separate video on the dark reactions, better known as the Calvin cycle. The dark reactions don't take place in the dark necessarily, they just don't require light, and so they're referred to as the dark reactions. So let's jump into the light reactions of photosynthesis, keeping in mind that we're in a plant cell, in a chloroplast, in a thylakoid membrane. So keep track of where all this is taking place. And we're going to focus first on what's called non-cyclic electron flow. So to understand the light reactions, we have to understand these special molecules called pigments. Pigments are what give things color, right? Because they reflect some wavelengths of light and absorb others. So if you look at a plant, I'm looking in my backyard right now, it's early spring, almost spring, very green. All that green that I'm seeing is all these pigment molecules. What are pigments? Pigments are molecules that have the ability to absorb energy, and in this case it's light energy. Three most common categories of pigments we see in plants are chlorophyll A, chlorophyll B, those are the two most abundant, and then a set called carotenoids like beta-carotene and others that have more brown, orange, yellow type colors to them. Chlorophyll A and B tend to be more greenish. These molecules have two main regions. They have a long hydrocarbon tail, and hopefully by now you're starting to see that a long hydrocarbon molecule is not going to do well in a cell. If it's in the cytoplasm or some other aqueous place, it's going to have to bury itself in the hydrophobic interior of a membrane. And so that hydrocarbon tail is going to lock these molecules into the thylakoid membrane. And then there's a more hydrophilic region, or somewhat hydrophilic region, that we refer to as the light-absorbing head. It's more specifically called a porphyrin ring. And this is where the variations are found between the different chlorophylls and the carotenoids. Now you might notice this has some symmetry to it that's interesting, and the symmetry is important. First thing you see is that we've got a lot of ring structures, and at the center we have a magnesium ion. The ring structures then, you notice, have alternating double bonds. Now you may not have taken organic chemistry yet, but I'll give you a little spoiler here. In organic chemistry, alternating double bonds create something called resonance. And resonance is essentially when the electrons from these double bonds have the ability to actually move across all of these atoms and be shared not only between the two atoms where they're drawn, but across all these atoms where we have the alternating double bonds. And so this, say, this electron pair represented by part of this double bond between this carbon and this carbon, 1 second may be around this carbon and maybe around this nitrogen later, and it may find itself on this carbon, fraction of a second beyond that, and this nitrogen after that. And so you've got this huge resonance, for lack of a better word, of these electrons moving and being shared across all of these atoms in this large multi-ring structure. Something about the physics of that, and this is beyond my pay grade as a biologist, you'll have to ask a pea chemist if you really want to get into the nitty-gritty of it. Something about the physics of this arrangement makes this like an antenna that just draws in, sort of, can absorb a packet of energy from sunlight, a photon of a specific wavelength from the sunlight. And we're going to see that this is essential then for capturing the energy of sunlight and using it to do chemical work that we think of as photosynthesis. Now, where do we find all of these? We find them in what's called an antenna complex. So an antenna complex is a very symmetrical array of pigment molecules embedded on the surface of what's called a photosystem. Photosystems 1 and 2 are going to be essential for understanding the light reactions, and we're going to dive into those here shortly. A photosystem is a complex of proteins embedded in transmembrane to the thylakoid membrane, with one end sticking to the lumen of the thylakoids and the other in the stroma of the chloroplast. And on the stroma face, we have the heads of the antenna complex, the heads of all of those photosynthetic pigments, and the tails are embedded in the hydrophobic interior down below. Now what happens is light will hit the antenna complex of both of these photosystems. We'll see that the reactivity actually needs to start at photosystem 2 and then move towards photosystem 1, but they're all scattered by the bazillions inside these thylakoid membranes in very close proximity to one another. And when a photon of light hits the head of one of these, I'm looking down here at the antenna complex, hits the head of one of these pigments, the energy is captured momentarily and then passed on to an adjacent pigment, and that's passed to another one that it's in contact with, and another, and it zigzags all around, transferring this energy until, through random movement, that energy packet hits a specialized region, a specialized pigment, in what's called a reaction center. We're going to focus on these reaction centers. So think of the antenna complex as a way of broadly spreading out, like I'm looking at my neighbor's roof over here, and they've got solar panels. You want as much surface area as you can for the sunlight to hit, and then you can capture as much of that energy. That's what these antenna complexes are doing. But it's not that every single one of these pigments can do something with the energy. They have to pass the energy along to these reaction center pigments. Once that energy hits the reaction center, that's when the magic happens. Let's take a look at that. See if I can get my, there we go. Okay. Nope, go back here. There we go. Okay, so we're inside a chloroplast, like you see up here in the upper left, and we're actually on the surface of a thylakoid membrane where the light reactions are taking place. A packet of energy, a photon of energy from sunlight, of a particular wavelength, hits one of these antenna complex pigments. It grabs the energy and hands it off to the guy that's next to him, who hands it off to the one that's next to him, who hands it off to the one that's next to him. The energy itself of this photon gets bounced around until it hits a reaction center. Now in photosystem 2, which I said has to start the process, not photosystem 1, photosystem 2, the reaction center is called P680. P680 takes those electrons and instead of, the energy, and instead of handing the energy off to another pigment, it takes a pair of its own electrons that are dedicated for this purpose, and it kicks them into a very high energy state. And that's what this yellow arrow here is representing. So a pair of electrons from P680 gets shot into a very high energy state from that photon of light. Now, we're going to see in a minute where those electrons go, but those electrons actually are going to get removed from P680. So now P680 is oxidized, and it becomes a very strong oxidizing agent. It's going to be very good at taking electrons. It really wants to replace those electrons it just lost. It's going to take them from somebody. Who does it take them from? Water. And the plant is saturated in water. The whole thing's saturated in water. None of this is going to happen. The plant's going to be dead if it doesn't have water. So water is freely available. Water is a terrible electron donor, but P680, when it's oxidized, is such a strong electron acceptor that it can just rip the electrons right off that H2O. It's called photolysis. Photolysis of water, splitting water into oxygen and protons by ripping those electrons off. And P680 is about the only thing naturally on the planet that can do this. We can force it to happen in the laboratory, but there are very few naturally existing molecules that are strong enough oxidants to rip electrons right off of water. Now, notice a couple things. This is where oxygen is made in the whole process of photosynthesis, is this first step in photosystem 2, regenerating electrons for P680. The other thing you should see is that this is essentially the reverse of what happens in cell respiration, where in cell respiration, oxygen collects electrons at the end of the electron transport chain and becomes water. Now we're just reversing that process, ripping electrons off of that water and generating oxygen, which is going to diffuse away, and we get to breathe it. Lucky us. Okay, so that's step one in this whole process of the light reactions in photosystem 2. I'm going to pause here, and I'm going to pick up in just a second. Okay, so what happens to those 2 electrons that are shot into orbit here, into a really high energy state? Well, they're grabbed by something generically called a primary acceptor. This is a protein molecule found in the thylakoid membrane. We know that the P680 primary acceptor is a protein called pheophytin. Pheophytin then, sound familiar if you've studied cellular respiration, is going to hand those electrons off to a shuttle called plastoquinone, sometimes called Q sub B. Plastoquinone then is going to shuttle those electrons to a cytochrome complex. The cytochrome complex will shuttle them to another molecule called plastocyanin, and then plastocyanin is going to hand them off to photosystem 1. Let's see if I can get this to look. There we go. It's going to hand them off to photosystem 1. Now, if you've studied cell respiration and electron transport chain, you know that in that scenario, the electron transport chain activates proton pumps, creating a proton gradient, and then that proton gradient is tapped into to drive ATP synthesis by chemiosmosis using an ATP synthetase. Turns out the same thing happens in photosynthesis. So these shuttles move the electrons from a high energy state to a lower energy state, and the cytochrome complex has proton pumps. And so the cytochrome complex is going to create a proton gradient across the thylakoid membrane. That gradient then is going to be used to power an ATP synthetase to make ATP. So remember, the 2 molecules we need from the light reaction are ATP and NADPH. So here's how the ATP is made. Now, where does plastocyanin go with those electrons? It hands them off to the reaction complex, the center of P700. Okay, so P700 then needs to be able to bounce those electrons into a high energy state for the second step, which is to give them ultimately to NADPH. Because after traveling from pheophytin all the way down through plastocyanin, they're in a low energy state. NADP plus can't take them yet. So they go to P700, which is part of photosystem 1. A photon of light will bounce around from one pigment molecule to the next until it hits a reaction center in P700, which is what we call it for photosystem 1. It gets knocked into a high energy state. A primary acceptor grabs it, and I don't have a name for you like pheophytin. It's actually pretty difficult to find anybody that names this primary acceptor. But there is an acceptor there, a protein in the membrane. It hands the electrons off through a series of carriers, including one called ferredoxin. And ferredoxin then hands it off to NADP plus reductase, which, as the name implies, reduces NADP plus by giving electrons to it, converting it to NADPH. And so the end result is photosystem 2 creates the ATP through chemiosmosis, and photosystem 1 generates NADPH. And we need NADPH and ATP to drive the Calvin cycle. So hopefully this makes sense to you. Camp out on this image for a while. Work your way through it. Sit at home alone and talk about it out loud if you have to. Call a friend in the class or meet up with somebody and go through it. Don't move on to the next video, which is cyclic electron flow, until this non-cyclic electron flow from the light reactions makes sense to you guys. All right, thanks for listening.